early.
Hello, everyone, and thank you all for joining us for our weekly Hanauma Talk seminar series presented by the Hawaii Sea Grant Hanauma Bay Education Program. My name is Gavin E.Y., and I am the Outreach Program Coordinator for the Education Program. First off, I would like to introduce Maya Walton, who is the Hawaii Sea Grant Program Leader and Research Coordinator. She acts as a liaison between various researchers, Sea Grant Extension faculty, and the University of Hawaii Sea Grant College Program. She also serves as a programmatic point of contact for several fellowships managed by Hawaii Sea Grant, such as the cadre of Sea Grant graduate fellows we are featuring here tonight. A few reminders before Maya introduces our speakers tonight. Please keep all your questions till the end, or if you do have a question during the talk, please type them in the chat room and we will address them at the end of the talk or in between the presentations. Also, please remember to turn off your microphone as well as your video during the different presentations. With that, Maya, would you talk a little bit about the Sea Grant Graduate Fellowship Program, as well as introduce our three featured speakers for tonight?
Mahalo, Gavin. And I see lots of people coming into the Zoom room. Aloha and welcome. Thank you so much for taking time out of your evening to watch these talks tonight. I also wanted to mention Tyler Lum is working in the background to do all sorts of magical things on Zoom, like let you in from the waiting room. So big thanks to Tyler, who's out there. I see thumbs up from Tyler. Thank you. Okay, aloha, I'm Maya Walton. I work at the University of Hawaii Sea Grant College Program as the research coordinator and the program leader there. And in the months of November and December, through the Hanauma Bay Lecture Series, we feature the work of the graduate students that we support through Hawaii Sea Grant, as well as the Pacific Islands Climate Adaptation Science Center, or PICASC for short. So I'm super excited. We have three great talks tonight from our graduate researchers. And again, these are graduate students that are supported through funding from Sea Grant as well as PICASC to complete research on oceans and coasts here in the Hawaiian Islands. And part of the training that these Sea Grant graduate fellows, these PICASC graduate scholars receive is training in science communication and in outreach. And we ask that these graduate students complete 50 hours of outreach a year. And one of the ways that they meet those 50 hours is through talks like these, talks to the general public through lecture series just like this. So I'm really excited to introduce our Sea Grant graduate fellows, or PICASC graduate scholars tonight. They're going to be sharing their research plans for the next year, and three speakers. So first we're going to hear from Sea Grant graduate fellow Paolo Marra-Biggs, and his talk is titled Samoan Hop Scotch, a Giant Clam Journey Across the Samoan Archipelago. The second talk will be from Casey McKenzie. Her talk is titled Dirty Groundwater, Linking Underground Pathways to Our Coastal Waters. And our last speaker tonight is Veronica Gibson, and her talk is titled Kona, where groundwater links biology, culture, and practice. So a couple of housekeeping notes. You'll see the chat. There's an icon probably at the bottom of your screen that says chat. And as we watch the talks from the different speakers, we encourage you to submit questions through the chat tool. And we'll take one question in between talks as we switch between speakers, but we'll have time for a longer Q&A at the end when we hear from all three. Okay, mahalo so much for joining us tonight. And I'm gonna hand the mic off here to Paolo.
Thank you, Maya. Sharing screen. Can everyone see that?
Yes, we can see. Looks good.
All right. Hello, and thank you for joining us tonight. from across the island, and I think there's a rare clam fan that's joining us from the mainland. My name is Paolo Marra-Biggs. I'm a second-year master's student at UH Manoa under my advisor, Rob Tonin, at the Hawaii Institute of Marine Biology. And tonight I'm going to be talking about my master's thesis research, playfully titled Samoan Hop Scotch, a Giant Clam Journey Across the Samoan Archipelago. So just to give some context, I'm going to introduce the place. The Samoan Archipelago is in the South Pacific, surrounded by a lot of water, about 2,500 miles south of Hawaii, and it's split into 4 main independent Samoan islands and 4 American Samoan islands, with a date line running right in the middle. So on a clear day, you can see your neighboring islands and see into tomorrow. It's quite cool. And my research is mainly based on the main island of Tutuila in American Samoa, where the National Park Service is. And these islands are amazing. They're drastic landscapes, volcanic intrusions that are just so beautiful. It's just amazing, spectacular backdrops. And the water themselves, the water surrounding these islands are quite amazing as well. Some of the coral species, of which there are 250 different species that are so well adapted to the rising water temperatures that they've been extensively studied from universities across the world, which kind of brings me to why I was there to begin with. These corals are very worth protecting. They're very important. And when I was down there, they were under attack. They were under attack by these sea stars that eat coral voraciously. They can eat about a coffee table worth of coral every single day. And they were getting out of hand. Their numbers were incredibly abundant. And so the National Park Service needed to take some action. And so we did a conservation through eradication project. And in about 5 years, we eradicated 27,000 of these sea stars, estimating with the predation rates that we saved about 300 football fields worth of this immaculate coral. Maybe asking yourself, why am I telling you this? Wasn't this about coral, or wasn't this about giant clams? Yes. Well, while we were studying these crown of thorns, we were being towed behind a boat, looking at all these coral reefs, looking for these bleaching marks or these predation spots. We got to notice some ecological imbalances in the reef. Specifically, there were very few of these beautiful critters. These are the giant clams. And across the world, there's about 12 species of them. And some of these bottom-dwelling behemoths can be 4 feet long and weigh 500 pounds. But they're slow growing, and they can grow to be 100 years old, making them the largest mollusks on the earth. And a fun fact, they're hermaphroditic. They can produce both eggs and sperm. And their tissues within them, they can hold algae to allow them to photosynthesize and generate energy from the sun. So we were being towed around this reef, looking for these crown of thorn sea stars, noticing some things were out of balance, and we started to inquire. We started to ask people, local elders, were they always this rare? And talking about the good old days, they would say, no, back in the day, they were abundant. You know, you could walk out on the reef and you could, not even with a mask, you could see their beautiful colors. You could pop them off and have it for dinner. And nowadays they're relatively rare to find. And so why do we care about clams being less and less common? Well, they're very important to the coral reef. They serve important ecosystem services for the rest of the coral reef animals. They're food for other fishes and invertebrates. And along with their photosynthesis, they can also filter feed. They can also take in water and clean it, allowing for clear water, which other corals like because they like to photosynthesize. Their shells themselves are huge, and they can create hard substrate of which corals can settle on and build more coral reef. I mentioned the symbiotic relationships with the menagerie of organisms that they have within their tissues, and they're also culturally important. A lot of the island nations use them in their traditional practices. But like I mentioned, they're of conservation concern. Because they're so big, they're stationary, they are dependent upon having other clams near them, which they can reproduce with. And the rarer they are, the less likely they are to be able to reproduce and fertilize those eggs and sperm. And they are becoming more rare. Their fishing pressures for them are increasing because some of their shells are being carved into beautiful artwork that can be sold for $100,000. And so people are going after them harder and harder, and more boats are going and harvesting them off the reef. And currently all species of giant clams are listed as vulnerable to extinction. So with the National Park Service in American Samoa, we wanted to look at the populations around the islands. So we focused in a small survey around the main island of Tutuila in just the national park waters. And we wanted to measure how many clams there were across multiple depths. So we decided to do some studies using transects that were 50 meters long by 2 meters wide to try to get an estimate, or a density estimate of clam populations in the main American Samoan Islands. And across 121 transects, we only found 100 clams, which means they're incredibly rare. And one cool fact that we found is they were growing down upwards of 100 feet deep, which, as you can imagine, the deeper you are, the darker it is. And if they like to photosynthesize, they're able to survive in some darker waters, unusually deep waters. As for which species we found, well, it was kind of hard to tell. Identification is kind of difficult with giant clams because there's a lot of characteristics that are shared between the species, and it's not as cut and dry. Some of the species share the same characteristics, and it's not as easy to tell when you're just looking at a giant clam because they're not obviously as clean as this. They're covered in coral and algae and wedged between rocks, and you can barely get a glimpse of them. But we decided to take a different approach. We tried to distinguish the species genetically. And so we wanted to go back out and collect a bunch of tissue samples, but unfortunately the weather and the seas did not agree with our field work. So we did some background research, and we found that in American Samoa, there wasn't a study done on giant clams since the late 1990s. And at that point, they were assuming all the species were the same. So we wanted to go and collect samples from the giant clams in American Samoa and identify them. And this study in 2017 was done on a genetic analysis across the Indian Ocean and the Pacific Ocean. And down at the bottom are all 12 species of giant clam. Here in the red arrow is where American Samoa is, and you notice that there isn't a black dot there, which means they didn't survey in the Samoan Archipelago. So we don't really know which species are present. We just are estimating based on this geographic range. So these are the 4 species that could be there, and we wanted to find out. So round 2, we went back and we did an island-wide survey across 3 different depths. And this time, because they were growing so deep, we wanted to measure their growth rates. So we had fixed sites. We pounded in rebar to mark these transects. And when we found a giant clam, we put a cattle tag on them. We wanted to find them in subsequent years. And we also took lots of photos, lots of notes to try to identify these species based on their nuanced characteristics. And we also collected those tissue samples. So here are some of the characteristics that we measured. The valve margins, different shell shapes and growth forms. Also trying to get photos of their in-current siphon tentacles, which you have to sneak up on these clams before they clamp closed, and it makes it very difficult for you to get a tissue sample after that. So you can see in the bottom right, after we size them, we kind of wedge something in them to keep them apart, and you can take a little slice of a tissue sample, of which we use to answer many different questions. So there's a theory in the American Samoan Archipelago that the ocean currents will potentially bring larvae or juvenile animals from the east to the west, from Rose Atoll to that main island of Tutuila. And using these giant clam tissue samples, we can ask the questions, are the clams floating from Rose Atoll and repopulating and replenishing the other islands? Or are they self-seeding? Are the populations independent of each other? And are they unique? So although we spent many years in the field, on the water with masks on, looking at this beautiful reef, the answers are in the genes. And so this is basically what my research looks like now. This is not malware. It's not a computer virus. Don't worry. This is the bioinformatics software the programs that we use to try to differentiate the genetic sequences that we find in these giant clams. And once we find out different unique sequences or signatures from all the species of giant clams, we can start asking the questions, well, what are present in American Samoa? And then once we figure that out, we can identify if these populations on each island are connected or independent. And depending on these results, that means the management actions would be slightly different. You'd have different regulations if they were all one population versus independently. And the more we know about these species, the better and the more effective we can be at creating regulations to protect them. So there's a lot of people to thank on this project. My advisor, University Sea Grant, all the agencies in American Samoa, and all my good friends at the National Park Service. I want to thank them, and thank you for listening. And to end on a comedic note, I wanted to tell a joke. What's a giant clam's favorite band?
What?
Pearl Jam.
Oh,
Pearl Jam. And if you have any questions, this is my email. Feel free to write me an email or write them in the chat.
Mahalo, Paolo. You're receiving virtual applause, real applause from me. Let's see, any questions for Paolo? Please feel free to enter them into the chat. And while we wait for questions to come in, I can ask a question. So you talked about the reproduction of giant clams. How does it happen? When does it happen? Tell us more.
Yeah, pretty cool. Because they're hermaphroditic, they can produce both eggs and sperm, but they cannot self-fertilize. So they actually produce one before the other as a better strategy to be able to spawn with other clams. Because you can imagine, you take turns, right? First, I'm the man and you're the woman, and then we switch, and I'm the woman, you're the man, and I produce the eggs and you produce the sperm. So you can create twice the amount of successful fertile zygotes. It's pretty neat.
And do all the clams spawn at the same time?
There is some studies based upon stress levels, so different temperatures can stress them out. So they have strategies based upon environmental conditions, but I think they do naturally, a time of year, they have a general spawning season.
Mahalo, Paolo. Okay, let's bring Casey's slides up. And while we bring Casey's slides up, we're going to take this other question from Kari. Kari, you want to ask your question, or would you like me to read it? I'll go ahead and read it. Kari asks, how long can eggs and sperm exist in the water column without a fertilizing opportunity?
That's a good question. I don't actually know that answer. I would imagine not very long. And the likelihood with the dilution of the oceans, the likelihood of them finding their conspecific to be able to fertilize is very unlikely. But that's a really good question.
Okay, audience members, keep those questions coming. We're gonna bring Casey up and start the second talk. Mahalo, Paolo. Casey, you might have to unmute yourself, but go ahead.
Thank you. Thank you so much. My name's Casey. I'm originally from Southern California, but I've been living on Oahu since early 2011. I originally completed my bachelor's at UH in global environmental science, and I worked under Rob Tonin. So that was pretty cool to hear about him again. When I was working with them, I completed my thesis looking at the compound impacts of climate change on crustose coralline algae. And the picture you see in the background here, I took while sampling some water chemistry for that project over on Coconut Island in Kaneohe Bay. I'm now a returning student to UH. I'm a first-year master's student in the Earth and Planetary Sciences department, and I just entered the program in August. So tonight I'll be presenting just some background information and methods that will be supporting my master's research project in the year to come. This research will be focused on linking underground pathways between contaminants derived from multiple land use sources and their submarine entry points in coastal waters. And it's not letting me, there we go. We know that groundwater contamination is often an environmental as well as human health concern, especially following major rain events when groundwater discharge is amplified. Scientists have identified multiple types of contaminants in this dirty water, including pathogens, pharmaceuticals, pesticides, detergents, and lots of other chemical cleaners. Some of these contaminants pose risks to human health, both for recreational users of our coastal zones and to those that consume other marine life, as well as the organisms that are part of the coastal ecosystem. Contaminated water also poses detrimental risks to our coral reefs. The picture you see illustrates a common issue in our reefs statewide, where coral colonies may be found covered by inundating algae. This algae is fueled by excess nutrients from wastewater and runoff. Basically, water that carries heavy loads of nitrogen from fertilizers and organic waste upstream is taken up by macroalgae that grow far quicker than the corals, and hence it's not uncommon to see our coral reefs smothered by this algal overgrowth. My project intends on helping us to define which human-derived sources might be contributing excess nitrogen that fuels this hyperproduction of macroalgae, along with other contaminants of concern that coincide with these nutrient sources. Previous research has confirmed that contaminated water not only infiltrates the ocean through runoff and sewer overflows, but it also intrudes the coastal waters from beneath the surface. So if you look at the image in the top right of your screen, you'll see that dye tracer studies were successfully used to show that wastewater effluent injected into the ground or released into cesspools disperses directly into the ocean in coastal communities like the County and the island. Other research has also been able to identify more contaminants that access the ocean via submarine groundwater discharge. This includes nutrients from agricultural uses and pharmaceuticals like caffeine, ibuprofen, and sewage. These contaminants I've mentioned, which are also commonly referred to as contaminants of emerging concern, or CECs, are defined this way by entities like the EPA due to their increasing detection in surface and groundwaters. Although we may not know how much of or how some of these contaminants impact the environment, their presence has become more common even at low levels in our waters, and have become a concern for what impacts they might have on aquatic life. My research will be sampling groundwater for some of these specific CECs as tracers that are unique to their land use sources. So here I'll be giving a brief visual description of what exactly submarine groundwater discharge is and how it differs from storm runoff. So we know that our surface environments are first to experience interaction from rainfall. Mountains, lakes, streams, vegetation, all life absorbs a portion of this rainfall. The excess rainfall then discharges to the ocean through streams and surface runoff, or it saturates the earth and recharges our groundwater. Because the recharged groundwater must first pass through the terrestrial environments before it's absorbed into the ground, this water directly interacts with potential contaminants in urbanized and natural environments. Contaminants may also be directly added into groundwater through effluent injection or cesspool leakage. This now contaminated fresh groundwater eventually does mix with the saline groundwater and exits through the seafloor as brackish submarine groundwater discharge, or SGD. For those of you that frequent the beaches, you might be more familiar with SGD in the context of coastal springs by experiencing patches of cold water while you're swimming or wading through the coastal zone. In the next few slides, I'll discuss some of the categories of contaminants among other CECs that I'll be examining in SGD for my research. The challenge we face is that there are multiple sources for which excess nutrients come from. And the nutrients that, that are displaced in excess promote excessive algal growth over our reefs. We can test for nitrogen, which is a primary component in nutrified water. However, if we are going to be able to define exactly where these nutrients are coming from, then we need to test for additional contaminant tracers as well. So what do we test for? We know there's different types of uses for land that may be sources of either natural or anthropogenic added nitrogen. For example, agricultural activities like croplands as well as pasture lands contribute nutrients, fertilizers, and pesticides to the groundwater. We can also consider on-site sewage disposal systems or septic tanks and cesspools that release partially or even completely untreated human waste rich in nutrients, pharmaceuticals, and personal care products directly into the groundwater as well. Leaky sewer lines and sewer overflows also contribute household chemicals as well as cleaning agents used in industry. These land uses will be our targets, and we intend to examine the hydraulic and chemical connection between these land uses and coastal springs by using pesticides and household-derived chemicals as land use tracers and measuring where the nutrients are coming from. Another class of contaminants we can look at are restricted use pesticides. Restricted use pesticides, or RUPs, are also commonly accessible herbicides like Roundup or glyphosate might be something you're more familiar with. Since RUPs are only permitted for use by licensed applicators, there's a limited volume of sources that could contribute nitrogen-rich groundwater that is also traceable with an RUP. So land use categories that use RUPs in large volumes are mostly limited to just agricultural use and landscaping entities like resorts and golf courses. Lastly, we can also test for industrial chemicals. These may most notably be found in floor cleansing detergents, like those used in supermarkets and retail stores. We may also be able to test for chemicals that are used in furniture polishes and finished products, like PDDEs or alkyl phenols. Since these facilities are usually on sewer rather than individual OSDS, identifying contaminants like these could help us differentiate sewage leaks from cesspool contaminated groundwater, as the cleansing agents are more likely to be found in sewer water. I forgot, there is one more last contaminant class we can test for is pharmaceuticals. Most notably, caffeine and ibuprofen are really common pharmaceuticals that we can find in nutrified groundwater that's affected by residential entities, such as cesspool leaks. So we will be testing for these as well. So overall, I want you guys to imagine a flow path starting from the mountains and going towards the ocean. And in that flow path, there might be nutrients from natural soil and pasture land use, pesticides from agriculture, detergents from commercial industry, caffeine and pharmaceuticals in our waste, and our sewage effluent. All of these potential contaminants all accessing the groundwater within the same flow path and coming out into our coastal zones via submarine groundwater discharge. This is the flow path that our project is aiming to identify by examining groundwater contaminants in submarine groundwater discharge. and in the coastal zones directly adjacent to land use practices, like golf courses, resorts, and any areas with a high volume of cesspools or effluent injection. Although previous research suggests that these contaminants have individually made their way from their respective resources into the groundwater, our ultimate goal is to use a combination of geochemical and pollution tracers to characterize possible flow lines and determining the types of land use categories that overlie them. The goal is to show hydrological and chemical connection between different land use categories and the coastal streams. So lastly, I'm going to describe some of the methods that we will be using. I haven't personally participated in this process yet, so bear with me. I'm a little rusty on understanding how it all works. But we will be taking samples at the shoreline at low tide in order to get the least diluted sample of groundwater from known coastal springs. The metal rod, called a push point sampler that you can see in the bottom left and right images, is an example of a sampling technique that's used to capture groundwater in the subsurface before it makes it into the ocean and gets diluted by mixing. Separate groundwater samples will be collected in multiple containers, as you can see on the screen. Radium is an SGD tracer through which we can quantify groundwater discharge rates, and we will also be using isotopes of hydrogen and oxygen-derived groundwater recharge elevation, and to help us understand the flow paths and where these land uses are accessing groundwater. The contaminants of emerging concern that I also discussed will be tested for, and so that's the bottle you see identified as CECs. We're hoping that in developing a framework that allows us to identify these connections between land use sources and the coastal waters, that we can provide this information to land use managers so maybe they can help better improve their methods in using whatever contaminant sources they're applying to their lands. I want to thank you all very much for joining us tonight and listening to my talk. I'd also like to thank my advisors, Henrietta and Kim, who've been very thorough in guiding me on how to research this material. It's all very new for me, and I'm looking forward to bringing this project to life so that way next year I can share my findings with all of you.
Mahalo, Casey. Thank you for your talk.
Thank you.
And giving the thumbs up. Oh, great. Awesome. So there is a question in the chat. So let's take this question from Anita. Anita, would you like to read your question out loud? Or do you want me to read it?
Sure. Sorry, my internet's not really stable, so sorry if I cut out. But hi, yeah, really great talk, Casey. I guess I've just been seeing a lot more studies going around on submarine groundwater discharge and cesspools around the main Hawaiian Islands recently. And I'm curious to see if you know if there's any statewide maps available that show exactly where these cesspool leaks are happening or the submarine groundwater discharges, and how are you deciding where to collect your samples?
That's a really great question. So there are maps on the state website. I can't remember the exact website URL right now, but there's a lot of GIS maps that have a collection of cesspools that are known around the state, and there has been research done identifying exactly where, like, known hotspots where SGD is coming out in the coastlines. I don't have that tonight, but I'm working on accessing those and learning how to use GIS so that way I can put a map together for my final project. And I will say that I do know that Hawaii, the state of Hawaii, has the most cesspools per capita, I think, than any other state in the US. So it's definitely a topic of concern. I have heard other talks about this as well. Our project is going to be looking at SGD on the Big Island off the South Kohala coast. As I said, I've only just started, so I'm just starting to delve into all of this. Thank you. That was a great question. I hope I answered it.
Mahalo, Casey, and mahalo, Anita, for your question. Keep the questions coming in the chat. We're gonna bring our next speaker up. Veronica's next.
I will share screen.
That sounds good.
Okay, does it look and sound okay for you?
Yep, we can see your slides and it sounds very good.
All right. Aloha, ahi ahi kako. Good evening, everyone. My name is Veronica Gibson, and today I'll be talking to you about Kailua, Kona, where groundwater links biology, culture, and practice. So first, a little bit about me. I am a phycologist, which means I study limu or macroalgae. So you can usually find me looking like something about the upper right-hand picture, like digging for algae on the reef. I'm a PhD candidate in the Smith Botany Lab. And usually I'm studying algae, but for this work, I'm taking you to my hometown in Kailua, Kona, because we really do live in this time where science is becoming informed by practice and by culture. So I'm really happy that I got involved in this work. So I started off studying the algae in groundwater, and I ended up with this project getting to study the ecosystems and why people care about them. So I'll be taking you to Kailua, Kona, which is on the leeward side of Hawaiʻi Island, the youngest of the Hawaiian Islands. This is a very dry side, so I'm super excited that we just got that great SGD introduction because Kona has no surface runoff. So virtually all of our groundwater or nutrients that enter the coastal ecosystem and our nearshore water sources are groundwater fed. So what that looks like is if you take a look at this bottom left-hand corner, this is Kiholo on the Big Island. And you can see all this new basalt. So we have very young basaltic rock here on the Big Island. And that means it's really porous. You can have holes like as small as your hair that water travels through, or you can have large lava tubes that literally carry tubes of water to the nearshore ecosystem and this coastal region. So what that looks like is that submarine groundwater can come out onto these reefs here, like Casey described, and change this ecosystem, and that'll be a groundwater dependent reef. We also have local ia, which are fish ponds that were engineered by Native Hawaiians to capture that runoff and to cultivate desirable species. And then we have where the water table extends above the rocks, but doesn't touch the ocean. So those are incline pools. So they have no surface connection to the ocean, but they do have underground mixing where that fresh water mixes with the salt water. So the pools can be, if they're closer and more connected to the ocean, very salty or very fresh as you move up the mountain. Or in the case of Kiholo, they just receive a lot of fresh water. So even though they're connected to the ocean here, there's so much fresh water coming out here that these ponds are very close to 100% fresh all the time. I think it's like 2 parts per thousand salinity. So once you get in the ocean in Kona, you'll see, like I did here, there's lots of productivity, there's lots of diversity and beautiful coral reef ecosystems. It's kind of world renowned for some of the best diving and free diving in the world. So today I get to talk to you about these ecosystems that I usually study algae in, but this is a project where we're using practice and lineal descendants and resource managers to inform how we're going to do our science in the next part of this project. So this portion of the research asks, how do people use, value, and care for GDEs, those groundwater dependent ecosystems in Kona, Hawaii? We looked at these 3 main GDEs that are found on the Kona coast. Local ia are those Hawaiian fish ponds where either the geology, like there's a wall built to enclose those springs and fish are cultured within that enclosure, or natural ponds are stocked with desirable species. So that's where we have the most human modification of the system. Oops, We also looked at Lokovai, Hopunapuna, or Kaheka. These all refer to incline pools, but that Hawaiian language really reflects the diversity of the different kinds of pools that we see. So they can have really different geology and different freshwater inputs based on where they're located and the freshwater they receive. So that means we see lots of different animals in them. Yeah. And then, so those are where the springs come out above sea level, but of course those springs still come out of that really porous basalt, even under the sea. So when we move into our nearshore ecosystem, we have muliwai, which is estuarine water. This is where those nearshore submarine groundwater discharge springs exit, and they can bring those nutrients to the shoreline that we talked about in the last talk. So usually I study how the algae lives here, but today we're talking about how people use, value, and care for these systems. So we're really coming back to the why I do my research and why a ton of people are working on these submarine groundwater discharge and the incline pools in the local IA. And that's really informed by place and culture. So the way that I investigated this was through a literature review and a series of interviews with resource managers and lineal descendants of Kona. And then I applied this Hawaiian Cultural Ecosystems Services Framework, which is really just a tool to explain the values that we get from these systems. And I use this framework developed by Pasqua in 2017, which is a really amazing framework to talk about this from a Hawaiian worldview and to describe it really in the context of Hawaii. So the history of these GDEs in Kona is that prior to European contact, these waters were the only source of fresh water for drinking, for bathing, for any need that you had, healing, spirituality, and birthing. So this is represented by this photo in the upper left. This is the Kahalalua Spring, actually from where I lived in Kona in Keauhou Bay. And this is the birthplace of Kauikeaoli, Kamehameha III in 1814, who is arguably the most famous ali'i throughout the islands. And this just really represents how the historical events and the history of Kona and the people of Kona were linked to these systems. Our communities were found where water was available, and the value of this water was so high that they were very intensely managed, and they were even viewed as ancestors. You know, they were revered. So after contact, people developed catchment systems and eventually pumping wells, and we moved away from this intensive management of these systems and this dependence on these systems. So during that time, there was a period of less management where ecosystems were overgrown, ecosystems were developed, and people weren't necessarily using or talking about or caring for these systems. This is represented by the next photo, Lanihau, where the old Kona airport was built over some incline pools in 1949. So there was some instances where, you know, this is the old Kona airport. These were some of the largest pools on the Kona coast, and they were built over. Following that period, what really started this, what comes next, is the Spirit Report was written in 1979, and this was really the first time that the Kona community came forward to say that we don't want these ecosystems to be developed. So actually, Koloko is in the background of this picture. There's a bunch of fish ponds there and a bunch of incline pools, and this was a very sacred place in historical times. So the lineal descendants and the people of Kona came forward to produce this report that was submitted to Congress in 1979. And they actually stopped development there, and they created a national historical park. And since that time in 1979, we've seen a huge resurgence along the Kona coast of different ways that the community has come forward to preserve these ecosystems, to preserve the knowledge associated with them. One example is Kohana Iki. So this is the bottom left-hand corner, I'm so sorry, where there's a luxury resort development that has partnered. It's a unique private-public community partnership. And the luxury resort actually has agreed with lineal descendants. They have a committee of planners to maintain these ecosystems in perpetuity. And they also maintain the shoreline and the camping there that is partially public. So Kona has really been looked to in recent years as a leader in being creative in how they're going to use this ancestral knowledge and their desire to preserve these ecosystems to create different ways to make marine protected areas and protected ecosystems. Part of this resurgence, you can see on the right is Kiholo. This is only one example, but a lot of fish ponds and incline pool managers along the coastline have started having community work days. And these are just amazing events where people can come together to learn not only the history and the culture, but to actually be a part of the restoration of these ecosystems and the culture associated with them. Oh, I should have said that Kona has the most incline pools of anywhere in the world per geographic area. And they're the only place that incline pools are found in the US. So it's a really unique and special place to study these systems. So just to explain the framework a little bit further that Pasqua developed, it divides the benefits that we derive from ecosystems into 4 categories. And these are overlapping, but they're just 4 broad categories that help us to explain these values. So Ike is knowledge from diverse sources, Mana is spirituality, Pilina Kanaka is social connections, and Ola Mau is physical and mental well-being. So now I'm going to take you through a series of quotes from my interviews that demonstrates these values. The first is Ike. To look at how water moves in the different vai from Mauka to Makai, how the cloud patterns in Kahaluu move and what captures it in the forest, the drip systems that start the process of creating water for Hawaiʻi. It is not rivers or streams, it is all underground systems. So we have a huge amount of water here, especially in Kahaluu. And this is one of those instances where our resource manager was also a lineal descendant. This kind of knowledge really, this quote was not unique in its broad sweeping observations that we don't always find in science so easily. We have this great wealth of knowledge in these people who work every day with these systems and make these huge connections, like from the clouds on the horizon to the water falling on the mountain and how that is related to the systems they work with. And I just include this as a reminder that we as scientists don't need to recreate the wheel. We need to look in the right places for this knowledge. And in that way, these people that we work with are hugely valuable to us. From there, more Ike, this same interviewee works with young kids, and she says, our young kids, once we train them, can go out into the beach area and they will actually present to the people sitting there. And it provides them different skills, public speaking skills, confidence, transferable personal skills. That is as important as teaching them how to take a YSI data water quality sample. So not only is she imparting this knowledge about these ecosystems, their ecology, their culture, and the science behind managing them, but these GDEs are also a tool to help these kids learn the confidence and the communication they need to express this to visitors and locals alike. So watch out for these kids that are going to come out of Kona in the next generation, because while I benefited from some of this growing up, it has gone so much farther than it was when I was there. They're giving so much great stuff nowadays. So then we move into Mana spirituality and Ola Mau well-being. What's happening with the individual as they're working with the ecosystems? When you take care of her, like the incline ponds and the vai that comes up there, it gives them opportunity in a non-threatening, non-judgmental space to be who they are. It allows them to drop some of the barriers that they create in their lives because of some of the trauma that they experience. And this, we discussed intergenerational trauma and individual trauma. So now we're seeing how the systems interacting with the mana of place is healing the individual. From there, we move into Pilina Kanaka, social interactions. On the community level, we have to bring up the awareness of our kids so they can have jobs in the planning department and even as judges and whoever it is, construction workers, that they have a deeper insight into how everything fits in and how what they do, how that will impact the source. And more about community work days, more Pilina Kanaka. People really want a place where they can be active and contributing to something, and I think this is the perfect site for that. And then moving into Ola Mau, the well-being that you gain from those work days. It's very significant to work in the pond, learn from the pond, and eat from the pond. It sustains and grows them. It makes that connection even more visceral. You can see how these categories blend because this is really crossing many categories, including Pilina Kanaka. So this final quote from a lineal descendant, I'll leave you with to think about the future of managing GDEs. You're not thinking in terms of years, you're thinking in terms of generations. To Malama is in not just washing the dishes on a daily basis, but it's sustaining not only the physical nature of water flow and our places in the hydrological cycle, but our relationship to the elements in the hydrologic cycle, including each other. So in conclusion, GDEs are valuable sites of biocultural restoration on the Kona coast that contribute to individual, community, and ecological health. Interviewees expressed that resilience needs to be a priority for GDEs, culture, and community. And just to talk to you guys a little bit about what's happening with this research next. So yes, we've identified that these are super valuable ecosystems. There's practices associated with the water that comes out there, and there's specific species that depend on them. There's so much that I want to share with you from these interviews that I don't have time to, but what's happening with this research next is we're working with a modeling team who's actually modeling what's happening with the hydrology. So the rainfall of Mauka, what happens to that water as it travels to the coastline, to these groundwater dependent ecosystems, how our pumping affects that, and also how pollution like the sewage we just learned about affects that water on the way to the coastline. So the next step is to learn exactly what do these practices need, how much water do we need, what quality of water do we need, and can we set some threshold, can we anticipate when these actions will impact those practices, and how can we prevent it? So with that, I want to say mahalo nui loa to my interviewees, my community partners, to Ikewai, I see a lot of you here, thank you, to the Kona community and my family, you're famous now, and to the Manoa community and Maury and my dad for pictures, and mahalo nui loa for listening.
Mahalo, Veronica. Okay, applause for Veronica. Great talk. I see all sorts of emojis and people clapping in their Brady Bunch chairs. Lots of good comments coming in through the chat, and I do see a question. So let's take this question. Well, some of these values, or I should ask, Robert Slanau, would you like to ask this question, or do you want me to read it?
No, I got it. So DAR is putting together management plans for all kinds of marine protected areas, and the values and things that you've collected are really important. And I'm just hoping that other people's work can be incorporated into the state 3030 plans. And I don't know if you've been approached yet, or if people are getting ready for how to integrate into that, but I'm told they want to have the plans already a year from now, so we all need to get cracking.
Yeah, so those did come up in our interviews. So some of my interviewees thankfully are involved with those management plans, but on my end, I am trying my best to get this information out as fast as possible, but we're working on writing it up. I hope it's ready in time for their plans. But great question, thank you.
Mahalo, Veronica. So we'll take questions for all three of our speakers now. And as a reminder, we heard from Paolo about giant clams, and then a great talk from Casey about submarine groundwater discharge and some things happening with water quality. And then our third talk tonight from Veronica on groundwater dependent ecosystems. So if you have questions, feel free to put them into the chat, or you can simply unmute your microphone and ask the question directly, or there's a raise hand function if you just want to raise your hand in the box. I'll try to scroll through and see if you have any questions for our speakers tonight. Okay, while we wait for those questions to come in, I have a question for Veronica. The groundwater dependent ecosystems, what's expected to happen to them under future rainfall conditions with climate change? What do we expect to happen to these really important systems along the Kona coast?
So you can stay tuned for the results of that work next year from Brittany when our modeling results come in. I guess I ran out of time, but we do have a few scenarios that we will run. So not only are we looking at how things are being impacted now, but we have a few scenarios that will run with changes in rainfall and other scenarios like changing septic systems to sewer and stuff like that. And then we also learned some new challenges from our interviewees. So sea level rise and tsunamis are also a big concern for the future management that isn't necessarily related to, well, still related to groundwater, but not solved by our study.
Awesome. It sounds like we have to have Brittany back for a talk on her work, her research soon. Any questions from the audience tonight for our 3 graduate researchers?
Hey, Paulo, so I know you answered it in the chat, but you want to address the question by Christina in case, people aren't reading in the chat?
Yeah, sure.
Yeah. So
Christina asked, how long do giant clam larvae stay in the pelagic phase before settling? And are there any current or physiological or oceanographic data or models that could help support the genetic data about the connectivity between islands? Very good question. And giant clam can stay stage for about 2 to 3 weeks. So they have all that time where they're in the water column drifting along with the main ocean currents until they find a spot and they settle on the bottom, and that's where they'll live the rest of their life. And as far as the oceanographic data, there are models. There's a study done by USGS. Because Rose Atoll is a nesting site for green sea turtles, they were very interested in how the current models will push the juvenile sea turtles who can't swim as well, where they'll end up settling or drifting along to. And so they found that there's a westward current from Rose Atoll towards the main populated Samoan Islands. And that's kind of where this theory is based. In terms of ground truthing it for marine invertebrates, that's what we hope to find out with our genetic studies.
Thanks, Paulo.
I think I saw Roberts raise his hand. Is that right?
I have a question for Casey.
Oh,
Hi, Robert.
So the testing you're doing for water quality is pretty sophisticated and really important to learn more about. But there's a lot of locations that would probably benefit from that kind of work. How can we take your findings and have perhaps a governmental agency pick up some of that information and start working with it so that we can have results in more places?
I think that's a really good question. I do agree that it's going to be applicable statewide for sure. I honestly, I don't know how to answer the question. I think that's something that will be, it'll be a networking opportunity that I'll come to learn more about as I'm doing doing the research. Pardon me, just for a minute. Grandma. Okay, baby. Pardon me. But yeah, I'm hoping that I'll learn more about how to network that and get our findings out more to government entities as we work on the project. Like I said, I just came in, so I'm just being inundated right now with all this information and trying to understand it better and how to organize and get ready to do the project.
Well, I can see you're good at multitasking.
My folks are watching right now too, so my daughter sees them on the screen.
Awesome. Glad to hear that there are family members watching these talks tonight. Thank you so much for joining us. Really appreciate it. Let's give our speakers one more round of applause, virtual round of applause. Great talks tonight. Thank you so much, everyone, for joining us. And don't forget, we have more talks next Thursday. So there is a new registration code that Gavin's going to send out for next. Oh, wait, is next Thursday?
No, Thanksgiving. So December 3rd.
Go ahead. Yeah, tell us about the schedule. So when are we coming back?
Yeah, so next week, excuse me, it is Thanksgiving, so we'll be taking a brief hiatus, and then we will be reconvening in December on Thursday, December 3rd, with another round of Sea Grant graduate fellows. So you guys can all have a great Thanksgiving.
That's right. Gavin, thank you for saving me. Happy Thanksgiving, everyone, and hope to see you back here on December 3rd. Mahalo.
With that, good night, and thank you.